I thought I would focus on the issue of variants and what the current situation is there, and other things. I think sadly we'll have to wait. So I chose this particular movie because I think it fit well with a quote that my colleague Carter Metcher made to us on an email string just within the last several days, saying that the emerging landscape of the pandemic now appears to be a variant soup rather than single variants like we have seen in previous waves, and kind of the madcap nature of the Marx Brothers and duck soup seemed like a good fit. So there's one study, I think in particular, that really highlights some of the issues that are driving this new phenomenon of variant soup that we're seeing. And this just came out very recently in preprint in MedArchive. It's from a team of researchers out of Beijing, looking specifically at immune escape and immunity changes in different emerging variants against previously vaccinated and infected with different variants. And what they find is among the recent emerging variants, there seem to be a theme of consistent mutations that are becoming dominant, and that these mutations all give these various variants the ability to escape neutralizing antibodies from vaccination and prior infection. They also, unfortunately, provide the ability for these variants to escape neutralization from the therapeutic antibodies that have been developed against COVID. And you can see that this quote from the paper says a high proportion of those variants display growth advantages over BA.5. And some of the recent variants that are popping up more and more are BA.2.3.20, BA.2.75.2, which we've seen here in the US, BR1, BN1, BJ1, BQ11. There's BXX and others, as you'll see, but that this phenomenon of rapid and simultaneous emergence of multiple variants with enormous growth advantages is unprecedented. So again, this, I think, highlights the idea that we're really looking towards a swarm of different viruses, or a soup of viruses that may be driving waves of COVID-19 in the future, rather than single variants that we've seen before. So in the study, what they did is they took the serum of patients who had been vaccinated with the Chinese coronavac. So again, take with a grain of salt, a little different from the mRNA vaccines we've had, less effective, but in terms of the relative efficacy across all variants, there should be some similarities in terms of how immunity has been generated. And in addition to subjects who'd been vaccinated with the coronavac, they were able to also identify subjects who'd been vaccinated fully, so 3 doses of the coronavac, and also infected with one of the more recent Omicron variants, so either infected with BA.1, infected with BA.2, or infected with BA.5. And what they see is, first of all, if you were vaccinated with coronavac, you had relatively high antibody titers against D614G. So that's the original virus out of Wuhan, with one slight modification. It was the dominant form of the virus in the spring of 2020. And so it's the virus essentially against which the vaccines were developed. So you can see very high, relatively high neutralizing antibodies against that virus. And if you were vaccinated and then infected with BA.1 or BA.2 or BA.5, you can see your antibody titer even went up a little bit. So getting vaccinated and then getting infected with any other variant gave you better neutralizing antibody titers against the original virus. Okay, makes sense. But now, as you look across the new variants that are emerging throughout the summer, you can, and now into the fall, you can see that many of these variants, and I highlight particularly here BA.2.75.2 and BQ1.1, the neutralizing antibody titer against those viruses is dramatically reduced compared to the original D614G virus. And again, all of the other variants that are been emerging recently, you can see have a significant reduction in neutralization compared to the original. So what we're seeing is as these viruses continue to emerge, we're drifting farther and farther away from the original virus. And that means that immunity against the original virus is becoming less and less potent, at least in the antibody neutralization titers, which again, we think in some ways correlates with overall immunity, but there are some, probably some significant differences, and it's not a perfect correlative immunity. The other thing to point out, and this is really interesting, is if you look at people who were infected with a certain virus after vaccination, and then look at their neutralizing antibody titers against that virus, it still isn't as good as the neutralizing antibody titers against the original D614G. And this is evidence that perhaps that idea of immune imprinting, or what people have colloquially termed original antigenic sin, may actually be something that we're seeing now in real life, and at least in these antibody studies, it appears to be the case. So you would think that if you were vaccinated and then infected with BA1, your neutralizing antibody titers against BA1 should be higher than against any other virus, right? Because that's the virus you were just infected with. The reality is, however, as you can see in panel D there, where that first orange arrow is, your neutralizing antibody titers against D614G are still higher than against BA1. And the same phenomenon exists for BA2 infection and for BA5 infection, which means that essentially, once you're originally vaccinated or originally infected with one strain of the coronavirus, and whether that was the original D614G, whether that was with Alpha or Delta, that's kind of the immune imprinting you got that's going to elicit the strongest response. And any subsequent coronavirus infection, or at least a SARS-CoV-2 infection, is still going to recall those memory B cells that fight against that original virus you saw. And that is going to potentially mean that B cells that would develop antibodies against the actual virus you were recently infected with, such as BA1, BA2, or BA5, is going to be less so. So really, what does this mean from a practical standpoint? It means that reinfection and potentially vaccination with new variant boosters may not elicit as good antibody response, at least to recent variants, as it does against some of the more original viruses. So it just means that as these viruses continue to evolve, we're going to continue to see potentially more and more immune escape. Recurrent infections are not necessarily going to protect you against the next version of the variant, and even vaccines may be losing efficacy. And we've seen that, although we've seen with repeat vaccination, we have maintained efficacy against severe hospitalization and death, and even recently some good data that you retain some efficacy against infection early on. So here's a couple of important quotes from what they described in the paper, that these divergent evolutionary courses seem to converge on certain mutations. So even though there's multiple different viruses, many of them share some of these specific mutations in the receptor binding domain, right? That's the business end of the spike protein that binds the human cells. And you can see some of those mutations there. And that these mutations seem to be related to immune escape, the ability to escape convalescent plasma, even those who had breakthrough infections and vaccination. They do comment that those two variants that I pointed out, BA275.2 and BAQQ1, seem to be the most antibody evasive so far, and that this phenomenon of humoral immune imprinting, or original antigenic sin, does seem to reflect itself in the neutralizing antibody titers that are seen in this study. So the other disturbing thing that was seen in this paper is these newer variants with these shared mutations seem to have dramatically reduced susceptibility to the therapeutics that we currently have. So as you probably know, as we moved through BA1 to BA2 to BA4 and 5, we ended up dropping a lot of the previous effective monoclonal antibody products that we had, and really have been left only with bebtelovimab for treatment and Evusheld, which is a 2 antibody cocktail for prevention. Unfortunately, as you can see down at the bottom there, any of those cells that are shaded in red means it essentially doesn't work. And so for BQ1 and BQ11, you can see that essentially absolutely no effect of bebtelovimab or Evusheld. And as again, we're moving down the evolutionary cascade here, you can see more and more these new variants are becoming resistant to those therapeutics because of these mutations, these shared mutations. Now, there is one product that still has some retained efficacy against all of these viruses, but it is still in development and currently not available. So if these variants indeed do take over, or at least many of them with these shared mutations take over, we may even lose our last therapeutic and prophylactic antibodies. So there's the BQ11 that I was discussing. So the other thing that I think is important to point out is even though these viruses are assimilating and accumulating mutations that allow them to escape immunity from vaccine and previous infection, it does not appear that this is tremendously impacting their ability to bind their receptor and at least gain entry into the cell. Actually, as you can see, as you get lower on the x-axis scale, that means your binding affinity is higher for the ACE receptor on the surface of the cell. So as you can see, most of, actually all of these new variants have increased binding of ACE receptors compared to the original D614G virus. So there's, I think, some theoretical arguments that as these viruses continue to gain mutations and escape immunity, they necessarily become less fit, less well suited to infect cells and cause disease. This would seem to indicate that that's not the case. These viruses are doing just fine binding and gaining entry into the cell, even though they have higher degrees of immune escape. So the conclusions of this paper talked about these additional mutations that they're seeing more commonly that allow them to, again, bind the ACE2 receptor while still evading immunity. And this phenomenon of convergent evolution, again, that lots of variants, but tending to congregate around these certain mutations, either in receptor binding domain or other parts of the spike protein, has been highlighted in a number of recent online fora and even Twitter. This is a figure I've seen bouncing around in a couple of places. I took it from Ryan Gregory's Twitter feed here, but again, I don't even know who actually originally came up with this because I couldn't find the source. Nevertheless, I think it's a good graphic showing that from BA1, BA2, now you've had this kind of proliferation of a large number of different variants that continues to grow more and more, but they all tend to center around some of these key mutations that you see here. And these are some of the most concerning variants that we're seeing recently. You can see that there's the XBB, which is kind of a recombinant hybrid called the Griffin, I guess, virus, but also some of the BM variants, BA2.75.2 that was talked about, BQ1 down at the bottom. All of these are kind of congregating around many of these common mutations, but there's lots and lots of variants now. And we're seeing this play out in Europe. As you can see now, Germany is clearly in the throes of a new fall epidemic wave, which is looking like it's going to be another severe fall and winter in Europe, as we've been discussing. But there's no clear single variant now that's dominating in terms of recent genomics. Sequencing data is generally lagging a couple of weeks behind. So we're really seeing what was present at the very outset of this more recent surge. But again, it doesn't look like there's any single variants that are dominating. And if anything, there are more and more variants being added into the mix and into the soup in Germany. We're starting to see this phenomenon in the US as well. Our BA5 dominance, which we have been seeing now for a few months, is starting to wane. We have some variants that are taking a larger chunk like BA46, but again, a lot of them are now just a wide variety of variants that we're starting to see come onto the scene. And it's unclear if if any single is going to drive an epidemic wave, or we're going to see similar to what we're seeing in Europe, this kind of soup. And so I think a helpful analogy that I thought of is just looking at it like river channels. So the evolutionary landscape of prior infections, which a year or two ago, if you had a prior infection with COVID, it was really with one or two variants, right? You were either previously infected with the original virus, or maybe you were infected with Alpha, or maybe, last year you were infected with Delta, but that was a limited landscape in terms of the number of different variants you could have been infected with and the number of vaccines that you could have had. Now, and that landscape drove the virus into very restricted evolutionary pathways. And so that's kind of like the Nile, right? You have one single big channel that's carrying all the water, and navigating that is pretty easy. But now you have a landscape where people could have been infected with Alpha previously and BA1, or Delta and BA2, or the original variant and BA4, and they could have been vaccinated twice with an mRNA vaccine. They could have been vaccinated with J&J and an mRNA vaccine, right? So these combinations now are almost limitless. And what you have is this incredibly complex landscape that's like the Amazon, right? Where you have channels and pools and backtracks and everything, where it's very complicated now to navigate and understand how this is going to drive the evolution of the virus forward, and where things are going to flare up. And unfortunately, I think that complicated picture is what we're going to see going forward. So I don't expect that we're going to have a single variant similar to what we've seen previously, nor do I necessarily think we need new variants, but the reality is the evolution of the virus is taking us that direction, and these new variants are getting more and more common mutations here that create this immune escape that is going to make I think the fall and winter, late fall and winter here look pretty ugly. So that's all I have, and happy to answer any questions.